So this is the start of a short PowerPoint lecture on the end of chapter 48 and a little bit of chapter 49, actually the section 49.2 on the brain. So in our last set of lectures, we left off talking about how a nerve impulse travels down one neuron, and particularly how an action potential is passed along the axon. And so now we need to take a look at what happens at the end of the axon. So how does it travel on to a different cell? So basically the impulse has to jump the synapse, and the synapse is the connection between the neuron and whichever cell it's passing the nerve impulse onto. So it can either be another nerve cell or it could be an effector cell, which is something that's going to have a response. So that synapse, that junction between neurons, has to be jumped in some way. So we have two ways that that's going to occur. We have either an electrical type of synapse in which the electrical current is going to flow from one neuron directly to another, or we're going to have a chemical synapse where a chemical neurotransmitter is going to carry the information across the gap junction to either another neuron or a different cell. Most synapses found, particularly in humans, are chemical synapses. But let's start, first of all, just defining what happens with an electrical synapse and where are you going to find these. So first of all, you need to remember that they are going to be rare, but they are going to occur when there is a need for a very quick response or a quick transmission, or when there's a need for a synchronized response. So first of all, before we define what that means, just a quick overview of how an electrical synapse is going to work. And essentially what it's going to require is that the neuron that is transmitting the impulse needs to be close to the next neuron that it's going to transfer the response to. And it's going to actually transfer that response through a gap junction, and particularly through gap junction channels that you can see here in this diagram. So the ions are going to flow directly from one neuron to the next through these channels and through this very close connection between the two cells. So that would explain how this can occur very quickly as opposed to a chemical synapse, which is going to still be fast, but it's going to have a little bit more lag time as the neurotransmitter passes through. So you can see here the neurotransmitter can be released through a chemical synapse here, and there has to be time for it to travel before it comes through the receptors, as opposed to this direct channel to channel passage of ions right through here. So an example in humans where that you would find this, you basically would find electrical synapses within the brain of a human. You would find them in invertebrates, in things like lobsters or shrimps when they need to have a quick response to get away for safety. So, but with a human, the need for a synchronized response is going to be more of the function that's going to require the electrical synapse. So for example, in the hypothalamus of the brain, you are going to find electrical synapses because it helps to have this very quick synchronization of a lot of different nerve impulses occurring, or cells firing action potentials at the same time, so that there can be a burst of, for example, a hormone secretion into the circulatory system. So that's where we would need that synchronized response in some sort of hormone release, that it helps to have it all released at the same time instead of dribbling here and there out. So now, focusing more on the more common chemical synapse, the things that are going to be happening at a chemical synapse are that the action potential is going to depolarize the membrane and open up calcium channels. And so calcium ions signal neurotransmitter vesicles to then fuse with the membrane. And then what happens is the neurotransmitter, once it's been fused there, is going to be released into that synaptic space, and then diffuse across to the next cell that's going to be getting the response. And so at that cell, there are protein receptors and ion-gated channels where the neurotransmitter is going to bind as a ligand. And then that will then start the signal transduction pathway into that new cell, either the impulse to action potential and then next cell to occur, or some other sort of response if it's a different, like a muscle cell or a hormone secreting cell. Here's a picture showing the chemical synapse muscle effector cell. So you can see here is our neuron here with the action potential shown with this red arrow. And then you can see the synaptic vesicles here that are carrying the neurotransmitter. So as the action potential is traveling here, we have the synaptic vesicles beginning to fuse with the membrane here at the bottom. And then we see, and that's a result of the calcium that is flowing into the axon terminal right here. So the synaptic vesicle is releasing the neurotransmitter. In this case, we're going to have acetylcholine as the neurotransmitter, because that is what is going to bind with the receptors on the muscle cells in order to get the muscle to start its action. So the nerve impulse in the next neuron. Okay. So basically what this picture is just showing how if we have the nerve sending the neurotransmitter across to another neuron, we're going to trigger the exact same action potential using the same type of ideas with sodium and potassium diffusing into and out of the cell. So the very same thing that we studied before in terms of how an action potential is traveling down the neuron is going to now begin in this new neuron that has received the signal. This is a very close-up, very cool picture of a postsynaptic neuron. And you can see those, these right here are the synaptic terminals of the presynaptic neuron. So the presynaptic neuron, again, as a reminder, is the one that's sending the message, and the postsynaptic is the one that's receiving the message. So postsynaptic potentials fall into two types. We either have an EPSP, which is an excitatory postsynaptic potential, or we have an IPSP, which is an inhibitory postsynaptic potential. And so those actually, if you think about it, are very straightforward and common sense. With the excitatory postsynaptic potential, we're going to facilitate and have our nerve impulse continue. So it's something that's really amplifying or passing on the same message. So that means we're going to have a depolarization in order for the resting potential of the new cell to turn into an action potential. So we're getting that resting potential over the threshold to start that nerve impulse in the new cell. In contrast, if it's an IPSP, an inhibitory postsynaptic potential, then we have the downside of our action potential graph. So we have that hyperpolarization occurring that is actually causing there to be no more action potential. So that means we have, if you remember back to our graph, hyperpolarization means we have a surplus of the potassium moving into the neuron, and so we have just even a greater resting potential, essentially. That has to, we have a bigger gap to get over a threshold to actually have an action potential occur. So this is just shutting down, it's inhibiting the propagation of that nerve impulse. So unlike action potentials, the postsynaptic potentials are graded. So if we think back to our domino idea, with an action potential, once it gets started, it goes. And it's just one strength. And so it really, there is not a different grade of response. It's just going to go at the speed that the axon can make it travel. But the postsynaptic potential is graded. And so, for example, a single excitatory postsynaptic potential is usually too small to trigger an action potential in that new neuron. So we need a number of these occurring at the same time, or having them occur in rapid succession. And that actually, the more that we have occurring, or the more quickly these EPSPs are occurring, the greater the response is going to be, and the more quickly we're going to get a response in that new neuron. And that effect, that rapid succession of excitatory postsynaptic potentials, is called temporal summation. So a summation, adding things up. So it's over time, temporal, the adding up of the effects of the terminals of this new neuron being stimulated by the neurotransmitter. So the same neurotransmitter can produce different effects in different types of cells. There are 5 major classes of neurotransmitters. We have acetylcholine, we have biogenic amines, amino acids, neuropeptides, and gases. So be familiar with some of these examples here: the acetylcholine, the epinephrine, the dopamine, and the serotonin, and what type they are. So acetylcholine obviously is an acetylcholine group. Epinephrine is an adrenaline or biogenic amines. Dopamine is also a biogenic amine, and serotonin is as well. There's a great table you can find in your book that goes over these major neurotransmitters and actually gives you their structure and what class they belong to, and where you would find them occurring and functioning in the body. So again, acetylcholine as a group is a common neurotransmitter in vertebrates and invertebrates. In vertebrates, it's usually an excitatory transmitter, so it's going to be passing on, facilitating the movement of an action potential. A biogenic amine are things like epinephrine, norepinephrine, dopamine, and serotonin. And you would find those primarily in the central nervous system, although you can also find them in the peripheral nervous system. 2 types of amino acids are known to function as major neurotransmitters in the central nervous system. And one of them you're going to come across, you find it more and more in the literature, and that's GABA. That's gamma aminobutyric acid. And also glutamate. Those are the 2 amino acids that act as neurotransmitters. Neuropeptides are relatively short chains of amino acids that can also function as neurotransmitters. Endorphins are an example of a neuropeptide. Opiates, this is where they actually work, and as you guys, some of you did your research projects on some opiates, you saw in terms of chemical or cell communication how they can bind to the same receptor and outcompete the normal neurotransmitter like endorphins.